Hello and welcome to another lecture. Today we are talking about photosynthesis. Let's recap what we talked about last time. In the previous two lectures, we talked about cellular respiration. Cellular respiration is sort of the opposite of photosynthesis, if you will. In cellular respiration, sugars are broken down to produce ATP, while in photosynthesis, ATP is produced and then also used to produce sugars, which can then be broken down in cellular respiration. So it's sort of a circular process between those two major processes, photosynthesis on the one hand, cellular respiration on the other hand. So today we'll talk about photosynthesis, but in the past we talked about cellular respiration and we said that it breaks down sugar and produces ATP and carbon dioxide, requiring oxygen. We said that oxidative phosphorylation occurs along the inner mitochondrial membrane. We had talked about oxidative phosphorylation being comprised of the electron transport chain and chemiosmosis. That was the ATP synthesis production. We said that during electron transport chain, electrons are passed along a series of membrane proteins, and each step is a redox reaction. The last step delivers the electrons to oxygen to produce water. And you'll see today that this is a very similar process that also occurs in the chloroplasts during photosynthesis. It's also called an electron transport chain, but it's slightly different. And I'll try to point out the differences. We said last time that during the redox reactions of the electron transport chain, hydrogen ions are pumped into the mitochondrial matrix, creating an electrochemical gradient. And also, this is very similar in the chloroplast. This happening in the chloroplast, not in the mitochondria, but there's also a hydrogen or a proton gradient that is being set up through this electron transport chain. The electrochemical gradient is used to power ATP synthase. Same thing in photosynthesis today. And then we had also spoken about fermentation, which is a different way of using sugar without oxygen. So fermentation uses glycolysis and omits the TCA cycle and oxidative phosphorylation. And then beta oxidation, I said, breaks down fats, feeding acetyl-CoA into the TCA cycle. And cellular respiration is regulated at multiple steps in the pathway via allosteric enzymatic activation or inhibition. So these last three steps are very different, but the first 6 steps, you should take the time after this lecture, go back and see how cellular respiration and photosynthesis are similar and where they are different from each other. All right, so let's take an overview of photosynthesis. You will see that photosynthesis is a set of 2 sets of reactions. So I say here 2 sets of redox reactions, and you'll see that these are all redox reactions, and it's really important that we understand what a redox reaction is in this context, because it's all about electrons being taken away from a compound and added to another compound. And when you think back about what a redox reaction is, it's the movement of electrons, right? It's the gaining of electrons, or it's the losing of electrons. So what's happening in photosynthesis in these 2 reactions is that we have a set of reactions called the Calvin cycle. And in the Calvin cycle, we need ATP, NADPH. That's very similar to NADH, which you learned about in the context of cellular respiration, and carbon dioxide. So those are the 3 things that it requires. And then it produces sugars or carbohydrates. And this ATP and NADPH that it requires, that comes from the other set of reactions, and that's called the light reactions. And so in the light reactions, energy from the sun is used to deliver electrons into this electron transport chain. And so it takes electrons, moves it along to a different compound, and each of those steps where electrons are moved along from one compound to another compound is a redox reaction, because by definition electrons are either lost or gained, and usually both at the same time, just from different compounds. So one loses, one gains, just like a redox reaction. So in that set of reactions, water is required. You'll see how water is actually donating the electrons. It also produces some hydrogens, that are very useful, and they were used as well in this reaction. And then it produces, in the end, you could say, as a waste product for photosynthesis, it produces oxygen, and then the electrons in the end, and I'll tell you exactly how, from the water are being used to produce ATP and NADPH, and that is then fed into the Calvin cycle where it's needed. So in the end, really all that is that needs to go in is the sunlight with the energy, water, which donates the electrons in the end, and carbon dioxide. And carbon dioxide is then reduced to sugar. So hydrogens are added onto carbon dioxide to produce the sugars. And so in the end, we have energy, water, carbon dioxide, producing carbohydrates, and oxygen. All right, so let's take a look where this is all happening. Of course, this is happening in the chloroplast, and if you remember, you might have seen something like this through the microscope. All of these little green dots are chloroplasts, and this would be like the outline of one particular cell. You have all the chloroplasts in here, and then if you look at a chloroplast in more detail, you can see that it consists of an outer membrane and an inner membrane. And then inside these membranes, inside this inner membrane, is another set of membranes. And that's going to be really critical for understanding these reactions. And they are called thylakoid membranes. They look like these stacks, these little stacks of things. And so they have a membrane as well. They are called the thylakoid membranes. And a stack of these membranes, they're called grana. And inside the membrane is a substance just like cytoplasm, except that it's not called cytoplasm, it's called the lumen. It's the inside of these grana, of these thylakoid stacks. And then on the outside of the thylakoid membrane, but still inside the chloroplast, is the stroma. And the stroma is sort of the cytoplasm of the chloroplast. Let me tell you one thing that I will come back to at the very end. The way that this looks is really like a cell itself. See, it has an outer membrane, it has an inner membrane, and it has membrane-bound organelles inside, right? Except that this whole thing is an organelle. So it almost looks like an organism inside a cell. And that is exactly what people think it actually originally was. This was a prokaryotic organism that was engulfed by another one and basically made to produce sugars for this particular cell inside the cell. So it's a cell inside a cell. And of course, over evolutionary time, eukaryotic cells, plant cells in particular, have evolved away a little bit of that. This is no longer an organism. It cannot live by itself anymore. But it's sort of the remnant of an engulfed organism that had its own membrane, its own sets of membranes, and its own set of organelles. So we'll come back to that. So for right now, let's look at this chloroplast. Let's remember that it has inside membranes and an outside membrane, and that this is the place where photosynthesis occurs. So let's come back to these redox reactions for a few minutes. So remember from last time that we talked about the fact that oxidation is the loss of electrons, and reduction is the gain of electrons. I gave you this mnemonic: oil rig, oxidation is loss, reduction is gain. So maybe that helps to remember this. And then I also said that for all intents and purposes, really in biology, it's easier to see that what a reduction is, because almost always that really means that it's a gain of hydrogen. So yes, it's a gain of electrons, but they are not in the formula, right? They're difficult to see, but the hydrogens are easy to see. And so gaining or losing hydrogens is easier to see. So let's take a look here at this overall formula for photosynthesis. Carbon dioxide plus water. And so we're balancing this with multiple molecules. These are 6 molecules of water and 6 molecules of carbon dioxide. And they're producing 1 molecule of sugar. So this is glucose. I'll show you another way of looking at that in a second, one that you have already seen before. And then oxygen. So there's 6 molecules of this O2. Now I'm asking you, which one is the oxygen, which one is the oxidation, and which one is the reduction reaction? So think about this a little bit. Take a look, see, is this oxidized or is it reduced? Is carbon dioxide reduced or is it oxidized to sugar? And is water reduced or is it oxidized to become oxygen? Well, what is it? Of course, this is a reduction reaction, right? So this is gaining hydrogens. Carbon dioxide is reduced to sugar. So when you hear that, and you'll hear it all the time when you hear somebody talk about these kinds of reactions, or when you read the textbook, that you have a reduction, that carbon dioxide is reduced to sugar. And then sugars can be oxidized back to other products, carbon dioxide, for example. So that would be the reaction in reverse. So and you see that, of course, that here are the hydrogens. That's what's gained, right? And the other thing that I want to point out is that these hydrogens that are clustered around the carbon, of course, produce exactly this carbohydrate, right? The carbon skeleton, 6 carbons, and then water around it, H2O, except we have this 6 times. So for each carbon, we have an H2O around it. That gives us the carbohydrate. I'll show you that in a second in a different slide. But so this is where we are. We are in the chloroplast. We have these extra membranes inside. We have redox reactions that are producing sugar from carbon dioxide and from water. And now let's go and look at these 2 reactions that I told you about, the Calvin cycle and the light reaction. Let's start with the Calvin cycle. Your book has this little icon here, and I think it's quite useful that you can see where in the cell you are located, except this is all the chloroplast, right? So there's a chloroplast, here's a grana stack of these thylakoid membranes, and the Calvin cycle is shown here in this cyclic set of arrows. Now think back to the TCA cycle, or the citric acid cycle. So citric acid cycle is a cyclic set of reactions, of chemical reactions, and that's the same thing here in the Calvin cycle. It's a cyclic set of reactions. It's a little more complicated than the TCA cycle, and so that's why your book only shows you very few real chemical names for it. There's quite a few more in there, especially this part down here, the regeneration phase is complicated. But what we can certainly learn from it is that it's in its conception very similar. There's a molecule that's the acceptor molecule. It then goes through a series of steps. During that series of steps, certain products are made, and the molecule itself is modified. So things happen to that molecule. And in the end, that molecule, this is the acceptor molecule, that molecule is being changed, things happening to it, and energy is, in this case, lost. Remember that in the TCA cycle, it's gained. And then in the end, something has to be regenerated so that there is a molecule that can, again, accept more of the input molecules. So with the citric acid cycle, we had an input molecule, and then we had molecules that were going through here. They were producing NADH, they were producing a little bit of ATP. Here in the Calvin cycle, our acceptor molecule is a molecule that's called ribulose bisphosphate. It has 5 carbons. That's all really I want you to know. It's a 5-carbon compound. If you know that it's called RUBP, that would be great as well. And if you know that carbon dioxide is added to this ribulose bisphosphate, that would be also good. So a 5-carbon compound is now connected with a CO2 that's a 1-carbon compound, right? So that part here, this reaction, this fusion of carbon dioxide with the ribulose 1,5 bisphosphate, that is called the carboxylation reaction because you're getting carbon to be added to this. And so your book writes here, the addition of carbon dioxide to the 5-carbon compound RUBP is catalyzed by the enzyme Rubisco. And that's another name you should know. Rubisco is the enzyme that catalyzes this step. So what does an enzyme do? An enzyme always reduces the activation energy. So it makes that reaction more likely to occur at cellular temperatures. And this enzyme Rubisco, the first thing is that's obviously one might see is that it has the same kind of letters in here. So RUBP, Rubisco, right? There's something in that name that we'll talk about in a little bit as well. So Rubisco is the most abundant enzyme on Earth. If you look at all the enzymes that are there, Rubisco is by far the most abundant because it's in all the leaves. And anything green on Earth, there's a ton of Rubisco that is allowing photosynthesis to occur, to this carboxylation to occur, the fixation, as it's also called, the fixation of carbon to an acceptor molecule, which is the RUBP, the ribulose 1,5-bisphosphate. So this is a sugar, you can say, a carbohydrate, and it has a chain of 5 carbons, just like glucose, except it only has 5. Glucose has 6, right? So it's very similar in structure to that. All right. Carboxylation, 1 carbon plus 5 carbons gives us 6 carbons, except that is really unstable. And so what's happening very soon is that it breaks into this molecule 3-phosphoglycerate, or 3-PGA. If you don't know that name, that's fine with me, as long as you are in Bio 111. Just know that this carboxylated compound is very unstable, and so falls onto these 2 molecules. And then you have then the reduction reaction occurs. So then you adding you require ATP for these reactions, and in the reduction reactions, remember that you are reducing carbon dioxide, right? So this is the molecule that eventually is reduced. And what is reduction really? Well, it's an addition of hydrogens, right? Reducing means adding hydrogens. So you're adding to the carbon dioxide now that it's stuck to ribulose 1-bisphosphate, but nonetheless, you basically keep adding hydrogens to carbon. So you're reducing that, and that's happening here step by step. You're adding NADPH. NADP plus is the remnant of that. And in the end, you're getting sugar molecules. Sugar molecules that can then be produced into glucose, that can basically be fused into glucose. So at this point, the addition of hydrogen costing us ATP, that produces carbohydrates. And they get siphoned off here out of the cycle, but not all of them, because some of those molecules need to be used to regenerate this acceptor molecule. So if you start with 1 carbon here, and you have 5 carbons here, now you have 6, and then it breaks into 2 times 3. And then out of these 2 times 3 carbons, eventually one of those 3 carbon molecules is extracted, fused with another 3 carbon molecule to have a 6 carbon molecule. Then the other 3 carbon molecules, and multiple ones of those are needed to regenerate this 5 carbon molecule. And you see that this is where it becomes complicated, right? It's not as easy to say, well, 1 plus 5 is 6, and then 6 divided by 2 is 3, and then 1 goes out and the other one is used for regeneration, because you're missing 2 carbons then, right? So you need a little bit more of this, and a little bit less can go out. There's a set of reactions where the carbons go through a series of fusing with each other, breaking in a certain way, re-breaking, refusing, until we have a 5 carbon molecule. All you need to remember here is that this is the regeneration phase, also costs some ATP. So if you remember from the Calvin cycle for Bio 111 that it has 3 steps: the carboxylation reaction, the reduction reaction, and then the regeneration reaction, that would be great. And if you know that ribulose bisphosphate is the acceptor, that it accepts carbon dioxide, and that in the end it produces a triose phosphate, a 3 carbon sugar, which is siphoned off, and that costs NADPH and ATP, that would be great as well. So that is what I would hope that you remember from this particular slide. And I have written down here the inputs, ATP, NADPH, and carbon dioxide, and the products, carbohydrate. So let's look at it in one other way, and that is, so I colored this in here. This is, of course, our reaction where we have carbon dioxide and water producing sugar and oxygen. And I colored this because I wanted you to see where the hydrogens in the sugar come from and where the oxygen in the sugar come from, and the carbon. So the carbon, of course, comes from the carbon dioxide that the plant takes in. And then the oxygen comes from the water that it also has to pick up, right? You're watering your plants, or it rains, and so then water, can be picked up by the plant. That is the oxygen from the water. And then the hydrogen from the water becomes the hydrogen in the sugar. And so then if you count this up, you have 6 CO2. These 6 carbons here are these 6 carbons. And then these 6 H2Os, that's of course 12 hydrogens overall, they become these 12 hydrogens here. And then these 6 oxygens are these 6 oxygens, and they become the 6 oxygens in the sugar. And then this oxygen here, that is what is liberated. These 6 O2 molecules become oxygen that is then in the end exhaled from the plant, right? It's given off. All right, so one last way of looking at it, I wanted to make sure that you remember, what glucose looks like and how this is a carbohydrate, carbon chain in the middle, water around it. That's why it's called carbohydrate. So here you have your hydroxyl groups along this chain of carbons. All right, so that is what's happening in the Calvin cycle, and all that happens in the chloroplast, and all that happens in, let me show you here, right here in this part of the chloroplast. That's the stroma, right? That's the plasma-like substance of the chloroplast. That's where it's happening, not in the thylakoids. It happens in the stroma here. Okay, so now let's just sort of take a step back and look at a chloroplast. This is an electron micrograph, so an electron microscope, very fine resolution. You can see a lot more than you can see with just a light microscope. And these strands here, these bluish strands, those are the thylakoid membranes. And then these blobs here, these yellow-green blobs in here, those are called starch granules. So starch is made from sugar. If the plant has a lot of sugar and it doesn't need all the sugar at night, for example, or while it's growing, to produce more energy than ATP, in the form of ATP, than photosynthesis is producing for it, and you haven't actually learned yet how it's producing ATP. But if it has extra sugar and doesn't need that to break it down to produce more energy, then it stores it in these starch granules. So they can look like this, and that's where you have starchy stuff. So that is one way for the plant to store the starch, and it can then break it down and use the sugar when it needs the energy. I'm going to show you this slide that I showed you in, I think it was lecture 2, just so that you sort of look back, make sure that you keep looking back at the previous lectures, because they do all kind of fit together and build on each other. So what I'm showing you here, this is the chloroplast starch granules. This is one I showed you in lecture 2. Again, this is the same thing here, our starch granules. But in the second lecture, it was in the context of the glucose molecules producing long polymers. So I say here glucose is linked to make polymers, starch and cellulose. And back then we talked about the alpha and the beta glucose. We said how they were linked, right? You can have multiple individual molecules, glucose molecules. They can make these ring structures from the linear structure. This is the same as this, except that this has now closed from here to here, made a ring structure. And of course, the way that this is written is it's omitting all the carbons, right? So here you see the carbons, here you don't see the carbons. Doesn't mean that they're not in there, they're just not writing them because it gets pretty busy then in this depiction. And what I was saying in lecture 2 is that the ring can close in such a way that the last hydroxyl group can either point up or it can point down. So it can point up here or it can point down. And when you put a lot of glucose molecules together with hydroxyl groups that are pointing down, essentially, then you get starch. And this is called a 1,4 linkage of alpha glucose monomers. This is an alpha glucose monomer, alpha meaning hydroxyl group is down. If you link all of those together, you get starch. Now, if you don't, if you happen to get, and this is 50-50 chance, if you're getting this after ring closure, then this sugar can be made into cellulose. It's still glucose, it's still the same monomer, but if you're linking it 1 to 4, of course, meaning 1, 4, that's the carbon, right? The number of the carbon in that sugar ring. If you're linking those together of beta glucose, then you get cellulose. And what is cellulose? Cellulose is, of course, what is in cell walls. So the plant can then use the cellulose to produce cell walls, or it can use it, if it's alpha glucose, it can use its glucose to produce starch. And then starch looks like this. Starch will be in the chloroplast until it's used. And then these are just the structures of how the starch can be either in this helical form, or it can be in the helical form, and then it can have branches as well. All right, so that's the end of the first step. So we now know how we can make sugar in the Calvin cycle reactions, all redox reactions, and in the end we are reducing carbon dioxide to sugar, right? Sugar is the reduced form, carbon dioxide is the oxidized form. All right, so now we want to go and we want to look at the second part of the photosynthesis reaction, and that is the light reaction. The light reaction is where you are gaining all this ATP and NADPH that is required for the energy that's needed in the Calvin cycle. All right, so let's look at that reaction. So here we have the light reaction, and I say here the light reaction is where the energy for the Calvin cycle is produced. So in order to get all that ATP, you're not going through cellular respiration to get all the ATP. Of course you could, but that would be kind of silly if you're making energy from sunlight, right? That's the point of photosynthesis. Then if you had to break down a bunch of sugar to make sugar, that wouldn't make much sense. So there is a set of reactions in the photosynthesis process that's called the light reactions, and they are really spectacular, if you will. I mean, from an evolutionary standpoint, this is something that has evolved, and of course that is the reaction, the main reaction that powers all life on Earth, because it produces chemical energy from light energy using carbon dioxide and water, and then just the sun, right? That is what makes all the energy, the chemical energy, all the food for all of the organisms on Earth. Without photosynthesis, we would be toast. And not even toast, because toast is made of carbohydrates, right? So let's look at how the light reactions how they work. So the figure here shows you basically what energy is. So it's called the electromagnetic spectrum, and it has high energy waves and has low energy waves. And so all kinds of electromagnetic waves are around, and there are those that are really low energy and have very long waves. They're sort of like very, very slow. If you think about a rope, and I give you the rope in your hand, and I hold the rope in the other hand, and then I just move the rope a little bit, just sort of one up, one down. There will be one very long wave, right? So just sort of one up, one down until it gets to your side. And I don't put much energy in it. But if I take that rope end while you're holding the other rope end, and I really shake it up really hard, you can imagine that the wave that I'm producing is very small. There's lots of ups and downs. And that is more energy rich because I had to put all that energy in to make the rope produce these waves. That's sort of something you can keep in mind as you're thinking about wavelength and energy richness of these wavelengths. So gamma rays are rays that are very high in energy. That's what you get from radioactivity. X-rays, of course, really high energy, that they penetrate your body so that you can look into the inside, through tissue. Ultraviolet light, quite high energy. That's what you can use if you want to burn your skin to a crisp, if you're going to a suntan bed. And then visible light is sort of in between, right? That is where the energy content of visible light is, the kind of light that we can see, and that in fact can be used for photosynthesis. And then on the other end is infrared, microwaves, radio waves. They still have energy, but they don't actually have as much energy as the visible light, and certainly less than on the other side. Okay, so all of these are wavelengths. Electromagnetic particles, waves. A physicist could explain that in all kinds of ways. For us, we just want to think about this as energy that comes to us through the atmosphere. So if we now just look at the visible light and just take that part of the electromagnetic spectrum, we see that these are shorter wavelengths, these are longer wavelengths. So there's more energy in the blue and the purple, and there's less energy in the red. And that light can now be absorbed. Any substance that is colored can absorb lights from within the visible spectrum. And that is what makes them colored. So if you have something that is green, then it's only green because everything else is absorbed by this substance. So the substance, if you graph it on the wavelength, this is the wavelength from 400 to 700, and this is the amount of light from this visible spectrum that gets absorbed, then you can see that a green substance will absorb quite a bit here in the blue and the purple, and so the blue-green, and then hardly anything in the green. So in the green, the greenish yellow, it's not absorbing it, it's reflecting it. And then again, it's absorbing it in the red and orange and the yellow. And the reason why it's reflecting it is because that's why we can see it as green, right? So if this was a blue substance, then how would the absorption spectrum look? Well, it would look such that it's not absorbing in the blue, it's reflecting in the blue, and instead it's absorbing in the green and the red. So something like that would be our absorption spectrum for a blue substance. And for the red substance, of course, it would be something that is absorbing the blue and the green, but it's not absorbing the red, it's reflecting the red, and that is why it is red to our eyes. Okay, so chlorophyll is green because it's absorbing blue and red light. And when I say it's absorbing that light, what it's absorbing is the energy. It's taking the energy out of the visible spectrum, out of the electromagnetic radiation that's of this energy content and of this energy content. So we'll come back to that in a second. I want to first show you how it's absorbing that, why it's absorbing that. And that is because it has a light absorbing molecule, and that is called chlorophyll. So you've probably heard of chlorophyll. Chlorophyll is a long carbon hydrogen chain molecule and has this porphyrin ring, it's called. Not so important that you know exactly what that's called. It's kind of complicated, it has some nitrogen in it. A lot of these carbon, hydrogen, carbon, and then carbon, hydrogen chains here, right? So these are bonds that go around here. There's the nitrogen as well. And then in the middle of the whole thing is a magnesium that is complexed to the rest of this ring. So, little side note here, when you have plants in your house and you give them water and you give them light, and after a few years, maybe it's a long-lived houseplant, sort of starts to become a little sad, and you think, well, I haven't fed it. I should probably give it some plant food. Well, what is plant food? Plant food is fertilizer, and fertilizer contains a bunch of things, but it also contains magnesium, also contains nitrogen. But it also contains magnesium, and the reason why plants need magnesium is to make chlorophyll. So if you never give them any magnesium, and they can't get any out of the soil because the soil no longer contains any, or you're not growing it in soil but on little beads, then that's the reason why a plant needs plant food. But other than that, this is almost all carbon-hydrogen chains, right? Long carbon-hydrogen chains. So this is the place where it's absorbing light. And if you are in organic chemistry already, which most of you aren't, but if you are, when you are, you will learn that these double, single, double, single, double, single carbon bonds, those are places where light can easily be absorbed by the electrons that make up these bonds. Remember that bonds, these lines here between the carbons, those are 2 electrons, or in the case of a double bond, 4 electrons that are being shared between those 2 atoms. Right? So those electrons, they contain a certain amount of energy. Remember that they're swirling around the nucleus. If you think about that model, that gives them a certain amount of energy. If you were to extract them further away from their circular place where they're zooming around the nucleus, that would give them more energy, right? So then they could fall back towards the nucleus. That's the energy that's inside a bond, if you will. Okay, so now something is going to happen to these electrons as they're swerving around the nucleus in this particular molecule, and this is how it's graphed. So I say here, chlorophyll can convert light energy into chemical energy, and your book shows you 2 ways of what can happen to electrons in that chlorophyll molecule as light hits it. So light hits it, you know that light has energy, light is warm, like sunlight is warm, heats things up. And so this graph, what it shows is the amount of energy that's in an electron in this chlorophyll molecule in these particular bonds that I was showing you. And so here's light, so this is wavy, right? So there's wavelength, and so there's some energy in there is what it's trying to show. And this electron is currently in its ground state. It's just swooshing around. It's just doing what it's normally doing in the dark, let's say. And so now there comes a jolt of energy. Light is turned on, the sun comes out, and this energy can be absorbed by these electrons. They can't be absorbed by any kind of electrons, but they can be absorbed very well by electrons that are in these double, single carbon chains, and particularly well in chlorophyll. So when it does that, the absorption of visible light by an isolated chlorophyll molecule, then it results in the release of heat, and this is supposed to be saying fluorescence, when the electron returns to the ground state. Okay, so what does that mean? It means that the energy level in the ground state, here's our energy content, the energy level is there, so there's a little bit of energy in there. But when it absorbs the energy that comes from this wavelength that falls onto the chlorophyll molecule, it takes the energy because the energy gives the electron sort of a jolt, and it pushes it away from the nucleus. And when it pushes it away from the nucleus, of course, that's negative and the nucleus is positive. There is attraction between those two, right? So it can't just do that. It can't just move away from the nucleus because of that attraction, of the positive and negative attraction between those two, the protons and the electrons. But if there's enough energy that comes from the sun, then that electron can sort of take a little detour. And that's what it does here, and this is what the graph shows you, is it suddenly contains more energy. So now it contains more energy, and now we call it an excited state electron. That energy in the electron has to go somewhere. It cannot keep it. Electrons can't hold on to that energy. So they give off the energy, and they have now 3 choices. Not that they're really making a choice, but there are 3 possibilities. The first possibility is that that energy is released in the form of heat. So one way how you might know that already is from sitting on a warm day on a dark surface, let's say out on the street where there's this black asphalt. Well, black substances, they absorb a lot of light and they don't reflect a lot of light. White substances, on the other hand, reflect a lot of light and they don't absorb much. But you can see that the absorption is not kept in the asphalt, but is given off as heat. You can walk on it and you can feel it, and you can see that it warms up when it's in the sun to more than air temperature. And it's because the energy in the asphalt molecules is given off as heat. Now that also happens in chlorophyll, and so heat can come off as well. There's another way how energy can be given off, and that is when it's absorbed as light energy. It can either be given off as heat energy, but it can also be given off as light energy again. And that kind of light energy is called fluorescence. So if you have something fluorescent, it's sort of shiny, right? It's kind of bright. You know, phosphorescent, fluorescent things, bright fluorescent green. Those are substances that give off the energy from the light that falls on them in the form of light again. And so that's what makes them fluorescent. And then so glow in the dark stuff is essentially fluorescent, except that there's a time lag between when it's giving off the energy, and that's called phosphorescent. But that's also a form of light that is given off energy that's given off in the form of light. And so when you do that, you can take chlorophyll, you can extract it, you can do that in the lab, and you can shine light on it. You actually see that it glows a little bit, but you can do that to any molecule. So all this is showing you is that you add light to a substance, it will absorb the energy, get the electrons excited, and then they give the energy again off from the electrons. They fall back to the ground state. So now enter the plant cell and the chlorophyll in the plant cell together with all the other molecules around it. There, what can happen when the light strikes it, this energy is not simply absorbed and then released as heat or fluorescence. It is released as chemical energy. So what it says here is that the absorption of visible light by an antenna chlorophyll results in the transfer of that energy to an electron in a neighboring chlorophyll molecule. So it can just bounce around that energy. So that energy can now go to this other chlorophyll, it can go to another chlorophyll, it can go to another chlorophyll. And rather than giving off the energy in heat and fluorescence, it just sort of keeps that energy around by bouncing it between molecules. Now, that can't happen forever, but it can go on for a little while. And in the meantime, these electrons can collect energy, that's why they're called antenna molecules, until they get transferred to it's called a reaction center. So those are basically special chlorophyll molecules where other things occur. And I'll show you what occurs in those reaction centers in just a second. But for right now, just know or try to remember that light energy is lost to the plant as heat or light if it's the solution in the lab, if it's not in the plant cell. But if it's in the plant cell, then the light can be kept within the plant as electrons that are energy rich. So within the real tissue, within the plant, within the rest of the organism, this can happen. And we'll show exactly why that happens and exactly what happens there in a second. So now we come back to these antenna molecules and that reaction center I was mentioning. So these light excited energy rich electrons can move to a different molecule in a series of redox reactions. And so when I say redox reactions, you already know it's something to do with electrons being lost or electrons being gained. And so if we look at this photosystem is just a set of chlorophyll molecules. You can see that the light can strike here and then it can sort of bounce around until it gets to this reaction center. It could also strike over here and then bounce around until it gets to this reaction center. And that electron now can move away and can go somewhere else. If it now goes to an electron acceptor, right, a molecule, different molecule, not a chlorophyll, that accepts the electron, then that leaves this with a hole of electrons. It's suddenly missing an electron, and so it has to get the electron from somewhere else, and it gets it from an electron donor. And so you can say that in this reaction, where an electron is given away to an electron acceptor, you have a loss of an electron. So the reaction center is losing an electron, and of course, losing an electron is a oxidation, right? It's an oxidation reaction. So the oxidation of the reaction center leads to the loss of the electron and the movement of this electron to the neighbor. Let's call it just the neighbor for now. And in order to keep the system stable, that electron has to be replaced by an electron from somewhere else. And so the reaction center now grabs an electron because it's so reactive, because it's missing an electron, it's grabbing it from an electron donor. And that electron donor can be another molecule that is on the other side, for example, of this molecule that is missing one. I'll show you exactly what these donors and acceptors are in a second. But this reaction center is now gaining an electron, and so now you have a reduction of this reaction center back to its normal state. So that means this reaction center can go back and forth between an oxidized state and a reduced state. Right? So all that means is that the electron is missing or has been replaced. Okay, so what is this electron doing? Where is it going? And why does it need to be replaced? So now let's look at this in a little bit more sort of overview way. We now have two photosystems next to each other. Right before I showed you this one photosystem, I showed you that the electron was either gained or lost. And so now what I'm saying is that this electron, when it absorbs energy, can gain energy. And so, it goes up in this energy scheme here. If you have energy on the y-axis and the reaction movement moving along in the photosynthetic reaction here on the x-axis, the electron is excited, it moves away, and what it can do through a series of steps is it can take this electron and move it to an NADP plus. So that's an NAD plus, just like the electron carriers that we saw in the mitochondria. It has an extra phosphate. That's really all that it does in chloroplasts. Otherwise, it's the same NAD molecule, it just has an extra phosphate. So is this the oxidized or the reduced form? Of course, this is the oxidized form, right? Because it doesn't have a hydrogen there. This is the reduced form. So if this guy here, NADP plus, is going to be reduced, it needs to gain an electron. Reduction is electron gain, right? So an electron gained, but we also said is also a gained hydrogen. So what it's doing is it's taking a hydrogen, it's taking two electrons, and then it produces NADPH. And NADPH, just like NADH, is a very high energy molecule that can be used to power other reactions. And so these other reactions that NADPH is powering will come to in a minute. So let's just pocket it. Let's just say sunlight hit this, bounced around, an electron was elevated to a higher electron level, energy level, and it was expelled from this photocenter, and then it reduced an NADP plus a hydrogen to become NADPH. Okay, so now there's a hole here, right? So we need to refill that electron because it was just expelled and now it's gone for all intents and purposes for the photosystems, for the chlorophylls, because it's now stuck in NADPH. So how do we replace that? Well, we can replace it by electrons from other photosystems or from other molecules close by, but let's just say from this other photosystem here. That photosystem, it can't just take the electron from it, and when that electron is missing, then that electron has to be replaced again by something else, and so it grabs an electron from someplace, and the place where it's ultimately the electrons come from, when all is said and done, and all these electrons have been replaced that flow into NADPH, where do they come from? They come from water. And water, of course, is everywhere in the cell, hopefully, if the cell is well watered. And when water is split, so there's an enzyme, your book doesn't talk about it, not so important right now. It's called the water splitting enzyme. It's really an easy name for an enzyme to remember because it splits water. So it takes the water and splits it, and it takes the electrons out of the water. These electrons then replace the holes in the photosystems. You know, they're not physical holes, obviously, they're just lacking electrons. And if you look at this, H2O, there is hydrogen in there. There's now a hydrogen ion, a proton at the end. So that is, of course, because its electron has been diverted, right? That electron goes in here. Now there's a proton left with a charge, and there's oxygen left. And your book does not do a great job of balancing this reaction. But if you had two of those, right, then you would have 4 hydrogens, and you would have 4 electrons, and you would have the 2 oxygens left. But it's really only important for you to remember that out of the water come some protons, oxygen that the plant exhales, doesn't need it, it's a waste product for the plant, and then the electrons. And the electrons can replenish these holes that I call them in the photocenter. Now, you also see that there is additional energy here, right? So it's not just the grabbing of the of this photosystem that liberates the electrons here, but there's also a little bit of a push for more energy from the sun that can do this over here as well. So let's just say there's enough energy around for these electrons to be expelled, and there's a need for the electrons to be replaced, and they ultimately come from water, and in the end they go to NADPH. And that's what I write over here on the side. So if you remember that for the next quiz or exam, that would be perfect. So these are all redox reactions. There are 2 high energy electrons from photosystem 1. They reduce NADP plus to H plus H plus to NADPH. The missing electrons in photosystem 2 are replaced by electrons from photosystem 2, from photosystem 1, of course. Let's just do that. Photosystem 1 are replaced by electrons from photosystem 2. The missing electrons from photosystem 1 over here are then removed from water molecules, and the split water molecules can't hold onto its hydrogens. They come up here, and then it produces the electrons, the oxygen, and the hydrogens. So that is what's happening during this transport of electrons. But there's more. There's a little bit more that happens, and that is very fortunate for the production of ATP. It's also kind of complicated. So let's look at this. What does that look like? What does that remind you of? I would hope that it reminds you of what you saw during the electron transport chain in mitochondria. Now, this is not the mitochondrial membrane. This is the thylakoid membrane. And to orient you, we have the lumen, so that's the inside of the chloroplast. So inside these stacks, right, inside the grana, inside the thylakoid membranes, that's the lumen. And then outside we have the stroma. So this is the plasma, if you will, of the chloroplast. So what I just showed you is all happening here. And so how is it happening? It's happening over here that water is split. We're having the oxygen and the hydrogen. We're having the reduction of NADP plus to NADH, and the reduction occurs from these electrons, and they require hydrogen over here that they take from the stroma, these electrons, NADP plus, and they produce the NADPH, which is great because that's energy. It's one form of energy. Now, there's a few more things that happen, and that is, you can see all these other blobs here, right? So these blobs are the photocenters, photocenter 1, photocenter 2, or photosystem 1, photosystem 2. You see how the light is it's basically pushing along these electrons, and then there are some substances on the way that the electrons are encountering. And when I say encounter, it's just, they're not just sort of swerving by them, but they are reacting with them in redox reactions. So The electron will reduce the substance, and then the substance will reduce the next substance, and then the next substance will reduce the next substance. At some point, they kind of run out of energy, and that's why they need the energy over here and the energy over here for the electrons to stay high enough in energy content to do these redox reactions. Now, these are all membrane proteins, and they're also membrane transporters. So we talked about membrane transporters in general, and we talked about them specifically when we talked about cellular respiration. And now we're talking about them again specifically when we talk about photosynthesis. So these membrane transporters, they can transport the electrons. So there's a reaction going on, but they're also pumping hydrogen ions from the stroma into the lumen. So from the outside to the inside. And as they're pumping these hydrogen ions, the concentration of hydrogen ions gets higher in the inside, low on the outside, and that sets up a electrochemical gradient, right? So this more positive charge here, less positive charge here, that sets up a gradient, an electrochemical gradient. Just like what we discussed before when we had hydrogen pumps. So this is essentially a hydrogen pump. And now we have another membrane. I haven't talked to you about this yet at all. We have just talked about this part so far. Now comes this ATP synthase, and that is a membrane protein, a transmembrane protein. So it spans the membrane, it goes all the way through the membrane, that allows the hydrogen to flow through, just like it does in the mitochondrion. And when it does that, also just like in the mitochondrion, it turns this, there's some sort of a molecular mechanism in there that you can think of just like a rotor, or like a blade of a rotor. And the hydrogens push this blade, and with every turn of that blade, the energy is the energy of the hydrogen movement is changed into chemical energy, because that allows this enzyme to put ADP and PI together, sort of click it together, and out pops ATP. So that's what's happening with this electrochemical gradient. The electrochemical gradient is set up, is used then to produce ATP by hydrogen flowing through the ATP synthase. And how do we get this electrochemical gradient? We get it by pumping of hydrogens into the lumen. And why do we get that pumped? Where do we get the energy from? We get the energy from the electrons that are doing redox reactions along these transmembrane proteins. And where do we get the energy for the electrons to move along? Well, we get that from the sunlight that is liberating the electrons and moving them onwards to the next. And then where do the electrons ultimately come from? Well, they come from water, where the water is split into oxygen and hydrogen. And so I have that all here for you. These are the 4 steps, but I would read the steps from here to here to here and then move over to there, rather than reading them in a linear fashion. So electrons reduce NADP plus to NADPH. Electrons are missing, they get pumped, and then they get eventually taken away from water. And then once this electron gradient is set up, all those hydrogens can flow through the ATP synthase and produce ATP. And now we have 2 things. Now we have from this reaction not only NADPH, but also ATP. And remember from the Calvin cycle that is, of course, what the Calvin cycle needs. It needs ATP and NADPH. Okay, so your book has these 2 slides, and I want to go through them. See, that's the same as what I showed you just now, but it focuses on something else. It focuses on electron transport and the synthesis of NADH, NADPH. So when you read that in your book, you will see that the absorption of light energy drives the linear flow of electrons from water to NADPH. That's this linear flow, right? That's what they mean with linear flow. And, we have the proton transport, and that is what this slide shows you. So we have the electron, during electron transport, protons accumulate in the thylakoid lumen, right? That's what I was telling you here, how that happens because of the electron flow, the linear flow. But what this what this shows you, what this sort of isolates this picture, is where the hydrogens come from. They come from the water, they come from the pumping, they all go through the ATP synthase out, and that is what's producing the ATP. So protons flow back into the stroma through the ATP synthase, driving the synthesis of ATP. All right, so with that, you have the main components of photosynthesis. Now let me say a few things and then we'll finish it up. So there is it's sort of an interesting relic from evolutionary history, and that is that it's a reaction that's called photorespiration. Photorespiration is interesting because that enzyme that I mentioned at the beginning, so this should look sort of familiar, it should look like the Calvin cycle. That's this Calvin cycle. We talked about the oxygenation reaction. I mentioned that that enzyme that puts the carbon dioxide on top of the ribulose bisphosphate is called rubisco. And rubisco stands for ribulose bisphosphate carboxylase oxygenase. So a carboxylase, when it takes this and adds carbon, then that's the carboxylase reaction, but it has a second reaction function. It can do two things. Most enzymes just do one thing, but this is an enzyme that can do both two things. And in fact, that's actually not so good for plants. So it can also have an oxygenation function, and that means that it adds oxygen rather than carbon dioxide. And when it does that, that's no good for the plant, because the plant wants to take carbon dioxide from the atmosphere and it wants to reduce that to sugar, right? Sugar with no carbon in it is no sugar, is nothing. So it needs to put in carbon dioxide. If it puts in oxygen, it's not going to gain anything. So what's it going to do with this compound that's now junk? Excuse me. So with this molecule that now has an extra oxygen in it instead of carbon, all it can do is it can do a salvage reaction. So it can try to make the best of it. And what it does is it splits the molecule. Remember that this is 5 carbons. Now you're not adding carbon, not a 6 carbon. So you can't do that you split it into 2 times 3 carbons like you do in the regular Calvin cycle. So what it does here, it splits off 3 carbons, a 3-carbon molecule, and does with it what it normally does in the Calvin cycle. Some goes out, some needs to be required for regeneration. And then the 2 carbons, remember this is 5, right? We had 3 that we take off, we use those, but the other 2, it tries to sort of put them back into this pathway. And it does that costing it ATP, and it splits off more carbon dioxide, and then the last carbon gets used. This is a complicated pathway. We'll talk about more about that in plant physiology if you take that class. But for you, all you should know is that when you have the input of oxygen through the oxygenation reaction of rubisco, that leads to carbon loss, because a molecule that's called 2-phosphoglycolate cannot be used in the Calvin cycle. The conversion of 2-phosphoglycolate into 3-phosphoglycerate, which can be used in the Calvin cycle, requires ATP input and a net loss of previously fixed carbon. And that's bad, right? So you if you're a plant, you try to fix as much carbon as you can, but suddenly comes an oxygen, destroys your molecule. Now you're splitting it into half, not quite half. You're using some of it, and the rest you have to sort of muddle around with to make sure that the rest at least can be used, and you can remake a regeneration of a acceptor molecule so that you can hopefully incorporate the correct molecule next time through that this molecule goes through the Calvin cycle. So I said this is a relic from evolutionary history. Why is that? Well, it's because when the Calvin cycle evolved in organisms, that was before plants really evolved. And that means there wasn't really any splitting of oxygen yet. So these reactions, parts of these reactions, didn't happen. They happened in an atmosphere where there wasn't any oxygen. So this really didn't happen originally because there was no oxygen around. Plants are really what put the oxygen in the atmosphere of Earth. So when I say here, when photosynthesis first evolved on Earth, Earth's atmosphere had almost no oxygen, and so therefore this oxygenation reaction was no problem. Now, is it a huge problem for plants now? Well, you could argue that, but can't be that big of a problem because they're still around, right? And in fact, they thrived even though they were putting more and more oxygen into the atmosphere. So I say here photorespiration is a nuisance, not a catastrophe. It leads to loss of efficiency. Here's the loss of efficiency, right? So you're costing it ATP, it's costing CO2. You're getting less of the carbon fixed. You're losing some of the previously fixed carbon, but you're not losing more overall than you gain. You just lose a little bit. You're just not as efficient. And I say here that photorespiration produces less ATP overall. So with the incorporation of oxygenation, you get less ATP overall. And the result is some loss of previously fixed carbon dioxide back to the atmosphere. So if you think about photorespiration, if you remember, it's because of oxygen. It's because Rubisco can do oxygen or carbon dioxide. If you remember that it's a loss of efficiency, those will be great things to remember. That should really be all that you need to know. You don't need to remember the names of these compounds. That's for next classes for you to learn there. All right, I want to show you this particular slide. Just tells you, you might think that a leaf is this amazing powerhouse that can turn sunlight into sugar, and it can, but it's really not that efficient. So if you think about the efficiency, if you calculate out the efficiency, there's all kinds of losses that occur. There's some of the losses from the energy that is converted to heat. There is 60% of the sun's output is actually not within the range of those wavelengths in the visible light that can even be absorbed by chlorophyll, right? So they're way out in either UV light, right? There's UV light inside what comes from the sun. There's some infrared. So that's all not even available to chlorophyll. Some of it is reflected and transmitted. Some is lost during carbohydrate synthesis, including from photorespiration. And so the maximum that people have calculated that you can get from the sun overall is about 4% that makes it into the energy of carbohydrates. So that's not very much, but on the other hand, it happens all day long, all the day, all the time. So has worked quite well for organisms, right? So you can definitely make a perfectly nice livable world from a 2 to 4% yield from this reaction, from the photosynthetic reaction. Now, having said that, one thing that you might be thinking about or you might be interested in for a nice project in the future is how can I harness the energy from sun using this fabulous system, but make it even a little bit more fabulous by tweaking it, by working with it, by putting it in some sort of other system that could increase, through engineering of some sort, the efficiency. Wouldn't that be great? If we could do that, then maybe we could reduce energy that we use from other sources like carbon, fossil fuels, and so forth. So the fact that there is still quite a bit of wiggle room of the energy that is being put in and the energy that we actually harness, or that the plants really harness for us, might mean that there is some way where we could maybe potentially even enhance photosynthesis. Just something to think about. All right, so the other thing I wanted to mention is that you might think, how did it evolve? How does photosynthesis evolve? And if you look at the chloroplast and you think about the fact that it has these extra membranes, and I said at the very beginning, it sort of looks like an organism within an organism. Well, that is exactly what people think happened in evolution. So, excuse me. In evolution, what people think what has happened is that a photosynthetic bacterium called a cyanobacterium had these photosystems 1 and 2, and it engulfed this. It was engulfed by another organism, and that organism then took its membrane. So it's this yellow membrane. If you're trying to push this through a membrane, and it sort of it doesn't rupture the membrane and go through, but it takes part of the membrane with it and sort of envelops itself with it. Then you have an extra membrane, and that's exactly what we see here, right? We have the photosystems in this green membrane, that is the thylakoid membrane. Then we have the outside membrane of what used to be this bacterium, that is that's the inner membrane of the chloroplast. Then we have the outer membrane of the chloroplast, which could be what is left over from the engulfing of this organism through this membrane. So this membrane, the yellow membrane, is sort of the leftovers of when the chloroplast was engulfed by another molecule. So that part is what I would like you to know, or at least have heard about. Endosymbiosis is what that's called. Photosynthesis in eukaryotes, such as plants, evolved by endosymbiosis of a light-absorbing bacterium. All right, and so now let me leave you with just with these nice slides from your book. I would really urge you to use the visual synthesis of photosynthesis and cellular respiration that comes with the end chapter assignments of your book for chapter 8. It connects in a very nice way photosynthesis, the formation of NADP, NADPH, ATP from the light reaction with the Calvin cycle, and then takes the carbohydrate that comes from the Calvin cycle, and that can be used in aerobic respiration, both in plants and in animals eating plants or plants using their own carbohydrates at night. And then here we can go again through the aerobic respiration pathway where carbohydrates are broken down in glycolysis that produces NADH, where carbon dioxide is split off, where we then go through the citric acid cycle, where more NADH is produced, where more carbon dioxide is given off, some more ATP is produced, and then the electron transport chain and chemiosmosis or ATP synthesis over here at the end. So look at this, compare that with what's going on in the plant. And then these reactions down here are essentially the reactions up here, but in a slightly reduced format, so that you can just sort of see the main inputs and outputs. I would very much urge you to look at this. You can look at it on the slide, but you can also use your book and zoom in and out so that you can see all these reactions and really try to understand that this is happening in different compartments of the same cell, if it's a plant cell, with the chloroplast over here and the mitochondrion over here. And that sort of connects all the parts that we've discussed so far in class. We've talked about aerobic respiration, we've talked about photosynthesis, but we've also talked about membrane transporters and why membrane transporters are important. We talked about we've talked about how molecules move through membranes and how membrane transporters are able to set up electrochemical gradients. That's all that this is, and that this is, right? It's an electrochemical gradient. And then we talked about the molecules and the chemical structure of the molecules very early on in the class. So now you can see how a compound like carbon dioxide can become a carbohydrate by the reduction, by the adding of hydrogens, so that it can become a sugar molecule. And so then we have a carbohydrate being produced from these individual parts, from carbon dioxide to a carbohydrate, and the carbohydrate then being broken down again to liberate the energy that was put into it, and the production of the waste products in respiration. So take a look at that so that you can sort of put it all together and hopefully get a good understanding of where things happen in the cell and how energy flows through living systems. So let's sum up what we talked about today. We said that cellular respiration breaks down sugar and produces ATP and carbon dioxide. We said that at the very beginning. We said that it requires oxygen, but the reason why I'm bringing it back here is because oxygen is produced by photosynthesis, right? It requires oxygen for cellular respiration, but oxygen is produced by photosynthesis. I said that photosynthesis is divided into two parts, the Calvin cycle and the light reactions. We started with the Calvin cycle. We said it uses carbon dioxide, ATP, and NADPH. It produces sugars. The Calvin cycle gets its ATP and NADPH from the light reactions. The light reactions are all redox reactions, just like the Calvin cycle reactions are. But in the redox reactions of the light reactions, we are taking electrons that we're splitting from water, and we are eventually reducing NADP plus to NADPH. Remember, reduction is essentially the addition of a hydrogen. That's what you see here. More hydrogens are pumped to the thylakoid membrane. That sets up an electrochemical gradient, and then that electrochemical gradient is used to produce ATP, and that ATP can then be used in the Calvin cycle, right, to produce glucose. So here we have a complete chain of events: oxygen use, oxygen production, ATP production, ATP use, glucose production, glucose use, and that's where you have producers, plants, and consumers, animals, using the energy that comes through the system, that comes into the system from the sun and the atmosphere through carbon dioxide and oxygen. All right, and so that was a long lecture, but that concludes that part of our discussion of major metabolic reactions in life. And we'll move on from here to cell divisions. I see you then. I see you in class before. Bye-bye.